Okay, so we have talked about the different components of a virus. You can tell me what's a capsid. You can tell me what's a spike. We have talked about the different ways that a virus can infect a cell. You can tell me all those different steps of uncoding or maturation or biosynthesis. You can describe to me a latent cycle and a lytic cycle. But now let's talk about what actually happens when that virus is inside of a host. So there are some latent viral infections, things like cold sores and shingles. This is when the virus is going to remain in a host cell for long periods of time. So you may be infected, you develop a cold sore, and then it is going to be latent. You're not showing signs of a cold sore, you're not spreading a cold sore, but it's still in your body. And then at a later time, something stresses you out. Maybe you're stressed out with school or you're sick, and that cold sore is able to come back out as a latent infection. There are persistent viral infections. This is when a disease process occurs over a long period. Something like measles can sometimes develop into this complication called subacute sclerosing panencephalitis. This is a brain infection that is going to be persistent, last for a long time, and often can be fatal. Cancers can also be sometimes induced by viruses. So all of us have genes in our body known as oncogenes. These are important during embryonic development. I imagine you started off as a fertilized egg, a single cell, and that cell had to be able to divide a lot to grow a baby that's big enough to grow. And then it divided a lot to grow into the adult that you are now. Your cells have to grow a lot whenever you get a cut or an injury to heal that wound. So oncogenes are very important because they let our cells grow when they need to grow. But when they don't need to grow, those oncogenes need to be turned off. So an oncogene, if it's turned on inappropriately, it causes a cell to grow. So that's kind of that onco or that cancer meaning of it. So if an oncogene is activated, a normal cell is going to be turned into a cancer cell. That's what it takes to create cancer. So an oncovirus or an oncogenic virus is a virus that can activate an oncogene. It can turn on an oncogene. Sometimes this will be seen as a virus going through a lysogenic cycle that inserts itself into a chromosome of a person in a way that turns on that oncovirus or turns on that oncogene. And so the cell starts dividing and becomes cancerous. About 10% of cancers are known to be virus-induced. So what is the difference between a persistent viral infection and a latent viral infection? And how can a virus cause cancer? Then to grow viruses in a lab, viruses have to be grown in living cells. So you can't just say, here's some agar, put some virus on it, and it's going to grow. To grow a bacteriophage in lab, you're going to have your agar. Then you're going to cover it with bacteria. So you cover your plate with bacteria. And then you can add your bacteriophage on top of that. And then you give it plenty of time for the virus to grow. And you'll be able to see these spots called plaques. So each plaque is going to represent a place where the viruses killed the bacteria. So wherever the viruses are growing, they're killing the cells and producing these little holes in the bacteria, known as a plaque. So it's a clearing in a bacterial lawn resulting from lysis by phages. Theoretically, each phage that gets into the bacteria is going to create a plaque. So you could count your plaques and know how many viruses you started with. But sometimes you'll end up with 2 viruses in the same cell. So you could have 2 viruses that cause the same plaque. So when we're counting them, we're not going to say there are this many viruses. We're going to say there are this many plaque-forming units. And in general, a plaque-forming unit will correspond to how many viruses you started with. To grow animal viruses in a laboratory, you're going to have to grow them in animal cells. You can't just throw them on a bed of bacteria. So there's 3 different methods. One would be living animals. This is really not a very well-liked way to grow viruses because animals are expensive and they're hard to care for. There's a lot of ethical concerns surrounding, living animals. It's not financially or morally or emotionally people's first choice usually. But living animals are the way to study immune responses of viruses, so sometimes they're used for that. Embryonic eggs, typically this is only used to grow vaccines when you're creating a modified virus to grow a vaccine. Cell cultures are the preferred method. So this is when someone has perhaps donated some cancerous cells to research, and those cancer cells can just be propagated continually in a culture tube. So it's more preferred, it's more convenient. It means that, you can take Christmas Day off without having to come in and feed your animals. So cell cultures are the preferred and convenient and cost-effective way and emotional, moral way to grow most viruses for research. We've already talked about cytopathic effects of viruses, but let's just review again. These are changes in host cells caused by a virus infection, and they can be used to identify viruses. You should be able to tell me several different types of cytopathic effects, things like transformation, things like syncytia, inclusion bodies. I think we discussed like 8 or 9 different cytopathic effects in chapter 15. Then, a couple of weird little things that kind of spur off from our topic of viruses. Plant viruses are things that can infect plants. They can enter through wounds or from insects where they've been biting on a plant. But separate from viruses are viroids. We have a lot of similar sounding names here. We've talked about virions, viruses, and now viroids. These are all very different words. Don't get them confused. A viroid is just RNA, all by itself, a piece of RNA, no capsid. But that RNA is infectious on its own. These are only pathogens to plants. They don't have any codes for proteins. They are just RNA. One example would be potato spindle tuber disease. Then prions. Prions stand for proteinaceous infectious particles. So they're infectious proteins. There is no nucleic acid with them. They can be inherited. So a gene for prion could be inherited and passed down. They could also be transmissible by ingestion if the protein is eaten, or by transplant if it is in a transplanted organ, or if it is contaminating a surgical instrument. These create spongiform encephalopathy. That's a fun name to say, spongiform encephalopathy. That means it infects your encephalum and it will turn it to a sponge, not an I can soak up knowledge type of sponge, but an my brain has turned into a bunch of holes in it, I'm not going to survive type of sponge. These can infect sheep where they cause sheep scrapie. They can infect cows where they cause mad cow disease. They can infect people where it can create diseases like Creutzfeldt-Jakob, Gerstmann-Straussler-Scheinker, or fatal familial insomnia. Here's your notes. You can pause to take your notes if you need to, but I'm going to go to the picture to talk about how a protein can cause a disease. So here's your happy little cell making, DNA, making RNA, making proteins, putting those proteins on the surface, doing everything its little brain cell needs to do. Then along comes this prion. And prion is like the bad, wild, crazy kid at school who, comes and convinces your kid to start doing drugs and having sex and all of the bad kid stuff, right? So it is going to come along and it comes up and meets up with your sweet little angel baby and converts your sweet little angel baby into one of the wild childs itself. So when the prion interacts with the normal protein, it creates another prion. So pretty soon it's converting all of the proteins into prions. Some of those prions can even be taken inside of the cell by endocytosis. And at that point, any proteins inside the cell that it's making, those PRP proteins, can all be turned into the prion proteins. So all of the surface, you're not going to have that normal protein expressed there anymore. You're going to have the prion. The prion is not going to function how it should be, which leads to forming those holes, those plaques along the brain that are going to eventually cause the disease. So tell me about what plaques have to do in studying viruses. Contrast viroids and prions. Tell me a disease for each of those. And how do plant viruses enter host cells? Okay, that's it for chapter 12. We still have some fun diseases to talk about in our 21 through 26. So I will see you later. Let me know if you have any questions. Bye.